Hello and welcome everyone. Before we start, I would like to acknowledge the Ngunnawal people and the traditional custodians of the land that we are meeting here today. And I would like to pay my respect to elders past, present, and emerging. And I also want to extend that acknowledgement to all First Nation peoples who are gathering here today. Right, it is my absolute pleasure to introduce Doa today. And as always, it's very difficult for me when I talk about my student during the final seminar. Doa is our homegrown. So she did her undergrad from UC, went on to do honors, did brilliantly, completed with first class honors. And then she came to do PhD with us, myself, Janine, and Ambi. And one thing I learned from her for being resilient. It wasn't easy for many of my students. In a PhD is a bumpy ride, but in addition to face COVID, it was not easy. But so my hat off to all of you, particularly Doa, being a full mother, looking after 3 children, and also COVID disrupted her work immensely. She had to recast her project and came up with the brand new chapter that she's going to talk about it today. So well done. And without further ado, I'll hand it over to you. Thank you, Doa.
Thank you, Tariq. I just want to say thank you to Tariq for saying all these nice words about me. And to be honest, I can't believe that I'm standing here today because 6 years ago, I did my honors here, and then I promised myself, that's it, never again. And here I am today presenting my final PhD seminar. So it just shows if you love something so much, it's hard to walk away from it. And if it wasn't for all of your support and encouragement, I could have not done it, especially my supervisors, my family, and my husband. So I want to say thank you to everyone here who came to support me and listen to my PhD talk, which is about subtelomere and chromosome evolution in amniotes. So let's start. So I will start my talk with giving a little bit of background about the chromosome, the subtelomere structure and function and characteristic. And then I will be talking about my aims. For each aim, I will talk about the method, result, and the conclusion for each one of my aims. So our DNA is packaged into chromosomes. The way it's packaged, so the DNA double helix is wrapped around histone proteins, and these histone proteins with the DNA, they form a structure called nucleosomes. So these nucleosomes is further organized into a zigzag string shape, which finally packaged into our chromosome. So most chromosomes have 2 arms, the P short arm and the Q long arm, and they are separated by a small structure called the centromere in the middle. At the end of each chromosome, there's a telomere, and then the subtelomere lies at the end near the telomere region between the telomere and the body of the chromosome. Before I start talking about the subtelomere, I will talk a little bit about the telomere. So the telomere is the end region that caps our chromosomes, and it's usually known as the cap of the chromosome. And as we grow older, our telomeres grow shorter. At the cell level, what does that mean at the cell level? It means that every time our cells divide, the telomere length gets shorter until it reaches a critical length where the cell will die. So one of the main functions of the subtelomere is the maintenance and the regulation of the telomere region. Also, it does a lot other more function, and one of the most important function of the subtelomere is the phenotypic polymorphism and diversity that provides to the genome and individuals. And this function will be the focus of my talk, and it was the focus of my PhD project. So talking about the subtelomere, for the structure of the subtelomere, the structure and the length of the subtelomere vary across different organisms. However, the main building blocks of the subtelomeres are repeated elements and genes. Although they vary greatly between organisms, there are some conserved elements that extend from parasites to human. So for the specific components of the subtelomere structure, the subtelomere is built of long homology blocks, genes, and a lot of repeated elements, including RNAs, helicases, transposable elements, and telomere repeats. So when they study the telomere region, the subtelomeric region, these are the blocks that I just mentioned. And in this phylogeny, each one of these circles, the red and the green circle represent the research that was done on the subtelomeric region in different organisms. And as we can see, most of the research was on yeast and parasites and in human. So there is a lot more unknown species, information about the subtelomere in a lot of species. So I will go over the little components of the subtelomere quickly. So before I start talking about my results. So the subtelomere structure, the first component of the subtelomere is the long homology blocks. And these are big and large blocks of DNA that can be duplicated in the genome. So they can either be duplicated on the same chromosome, or they can also be duplicated in other chromosomes. Next, the extracellular genes. The genes that are found in the subtelomeric region are mainly genes that the subtelomere or the genome used for interacting with the environment or for adaptation. And in the human, few gene families were found in this region, including the olfactory genes, zinc finger proteins, and the immunoglobulin genes. Next, ribosomal DNAs. The ribosomal DNAs is it's an array of genes. These genes are repeated next to each other, and they encode for the ribosomes, which are the protein factories in our body. As I said, they are ultra conserved or highly conserved from prokaryotes to eukaryotes, and in yeast and insects they are found in the subtelomeric region. Then the tandem repeats. So the tandem repeats is one or two or more DNA base pairs that are repeated multiple times on the genomes, and they have to be repeated near each other to make a tandem repeat. Usually these tandem repeats are different from human to human, so they have variable numbers, and that's why they've used them in DNA fingerprinting and in breeding and genetic diversity studies in wild animals and domestic animals. So usually these repeats are, as I said, variable between humans, but sometimes if they go over a certain length, they start causing diseases such as Huntington's disease and Fragile X syndrome. Helicases, specifically the RIC family, these are they call them the guardian of the genome. They're very important for the stability of the genome. They've discovered 3-5 families in humans, and any disruption of the families of these RIC helicases, it can lead to a cancer predisposition, premature aging, and genome instability. Transposable elements, as you can tell from their names, they are DNA sequences that can jump or move in the genome from one position to the other. There's two classes of transposable elements in our genome, and they consist 50% of the human genomes and up to 90% of the corn genome. Finally, the telomere repeats. As I said at the beginning, telomere repeats found at the end of each chromosomes, but sometimes they can be found in other places in the genome. And then when they're found at other places other than the telomere region or the end, we call them interstitial telomeric repeats or ITS. And they are found in the subtelomere in human and yeast. So given the dynamic nature of each one of these components of the subtelomeric region, some of them are variable between different individuals, and some of them can jump from one region to the other. That makes the subtelomeric region a very dynamic and highly unstable region. Also, there are other things that contribute to the dynamic nature and the instability of the subtelomere, which includes the double-strand breaks and high rate of recombination, which can reach in some cases to 160 times in comparison to the rest of the genome. And all these instability can cause rearrangement, duplication, and deletion in the subtelomeric region. So these dynamic nature or instability of the subtelomeric region have consequences, and these consequences can be positive and negative consequences. So on one hand, it can cause as I said at the beginning, polymorphism and diversity between individuals and in the genome, but also they can cause disease such as cancer and infertility. So these are the short-term, and at the long-term consequence of this, the dynamic nature, it can lead to the adaptation and speciation that can cause between individuals and organisms. So now that we've learned about the different subtelomeric structures, each one of these, which are found right at the top of this phylogenetic tree, we can see that these different components have been studied in the top there, the yeast, and the bottom in Plasmodium and parasites. And out of vertebrate, most of the research, the only research that was done about the subtelomere was in human. So as you can see, there's a big knowledge gap that we still need to fill, which include all the empty circles in this phylogenetic tree. So in my project, I've decided to look at the subtelomeric region or these components in amniotes. So in details, the three different representative species of amniotes are, or groups of amniotes, mammals in the top circles, reptiles, and then birds. So the overall aim of the project is to look at these different components of the subtelomere structure and rearrangement in amniotes. And in my first chapter, I will be characterizing the subtelomeric region in the Tasmanian Devil, which represent mammals or marsupials from mammals. And then in my second chapter, I will be looking at the evolutionary dynamic of some of the subtelomeric repeats in birds and reptiles. And finally, in the last chapter, in chapter 3, I looked at the subtelomere genes distribution between reptiles and birds. So for the first chapter, I characterize the subtelomere in Tasmanian Devil. And as we all know, Tasmanian Devil is a marsupial, Australian marsupial from the dasyurid family, and it's an endangered species due to two facial tumors, the DFT1 and the DFT2. So the Tassie devil have interesting telomeres. They have different length of the telomere between different chromosomes. So some of them have extra long telomeres and some of them have normal or shorter telomeres. And this is a characteristic of many members of the dasyurid. So they believe that the telomere dimorphism is involved in the development of the DFT1 and the DFT2. So I've decided to use, and because it is one of the best assembled, it has one of the best assembled genomes out of all marsupials. So I'm using the Tassie Devil to represent the marsupial group in the mammalian group from Amniotes. So the method that I used for this chapter was mainly bioinformatic tools. So I've used Ensemble and NCBI to extract the DNA sequences and the genes and the GC contents from the databases. And then I used the plots from both NCBI and Ensemble to find the precise position for the gene, RDNAs, and the helicases. I used a tool called Cluster Omega to do the alignment for the segmental duplication. And then finally, I've used the repeat masker to look at the tandem repeats, telomere repeats, and transposable element in the subtelomere of Tasmanian Devil. So before I start analyzing all of these components, I wanted to know what is the length of the subtelomeric region. So the subtelomeric region can be different from one chromosome to the other, and it also can be different from one individual to the other, because of the repeats that can be different in length, and because of the transposable element and the segmental duplication that different between individuals. And usually in human or other organism, when they study the subtelomeric region, they take in consideration the abundance of segmental duplication and the GC content of this region, which is supposed to be higher than the rest of the genome. So in human, when they do the analysis for the subtelomere, usually they do for 500 kb. But for me, because I'm not sure what is the exact length of the subtelomere in the Tasmanian Devil, I decided to do the GC content for the whole region. So I did it for the 5 MB and for the 1 million base pair, 500 kb, and then for the 1 MB. And then I did it also for the whole genome, and I compared the three. So as you can see from this figure, I looked at the GC content in all of each one of the chromosomes and in the total of the genome. So the blue one represents the 500 kb region, the 1 MB is in orange, and the total chromosome is in gray. And as we can see here, we always had highest GC content in the 1 MB region. So I've decided to do the analysis for the 1 MB. This might suggest that the subtelomeric region could be longer or double the size in Tasmanian Devil in comparison to human, which is interesting because they also have extra large telomere. So it kind of making sense. So for the GC content, we did the analysis for the subtelomeric region, and then we did for the rest of the genome in the Tassie Devil. And when we compared it to the human, although the Tassie Devil and the human have different GC content in their genome generally, the subtelomeric region for both have very similar GC content, which is interesting. Now that I did the telomere length, and I know that I did the analysis for the 1 MB, so I can go with you through the results that we've extracted for the Tasmanian Devil for each one of these components. So I did the analysis for all of these components in each single of these subtelomeric regions from both arms of the chromosomes. And we'll go through the result together. So for the first one, we've identified 16 segmental duplication in the subtelomeric region of Tasmanian Devil. Most of these segmental duplication were duplicated on the same chromosomes, except for 2 of them. They were duplicated between chromosome 2 and chromosome 5. Then the extracellular genes, 214 genes we identified in the subtelomeric region in Tasmanian Devil. 21 of these were uncharacterized. And the ones that are highlighted in blue are common families between human and the Tasmanian Devil. The tandem repeats, to be able to do the tandem repeat analysis or the repeat analysis in the subtelomere, we had to do the repeat analysis for the whole genome. And then we did the tandem repeat analysis for the subtelomeric region, and we kind of filtered the common repeats between the whole genome and the subtelomeric region. So this area, and we ended up with 73 unique subtelomeric repeats. These repeats can be very informative in terms of the evolutionary timeline of the subtelomeric region in Tasmanian devil and other marsupials if they're conserved there. The telomere repeats. We found telomeric repeats in the subtelomere region of the Tasmanian devil at most of the chromosomes. However, the longest one, which almost double the size of the rest of the repeats, is in the P arm of chromosome 2. Transposable elements, 5 families were identified in the subtelomere of the Tasmanian devil, and they belong to the 2 main classes that I've talked about at the beginning of my talk. So now the green ones, all the ones that are highlighting green are the components that we found in the subtelomere of Tasmanian devil, and the ones in red here, we didn't find them in the subtelomere of the Tasmanian devil. So they're there in the genome, but not in the subtelomeric region. So this is just an example from the PM of chromosome 2 of all the components that I found in green. Those are the ones that we didn't find in the subtelomere, and that's how it would look like on the end arm of the chromosomes. That's the organization across the whole length of the subtelomere, with which include the repeats at the bottom, the segmental duplication, the ITSs, the genes, and the transposable element family. So now, what does that all mean in the bigger contents of my project? So what I've done is selected the Tasmanian devil to represent marsupials, and now after characterizing of this component, I now can fill all these empty gaps. So we know that the ones in green are found in the subtelomere of Tasmanian devil, and then the ones in red are not in the subtelomere. And if you can see from here that the structure of the subtelomere in the human and in the Tasmanian devil is generally similar to each other in terms of the different components, although there were some differences in the amount of these components and their organization, but the general structure was conserved between human and Tasmanian devil. Now, moving to the next 2 groups of amniotes, birds and reptiles. And in this chapter, I characterize the repeats of the subtelomeric region in both of these groups. So birds and reptiles are very highly diverse groups of animals, very high number of organisms. However, their genome are very similar to each other. The karyotype or their chromosomes are mainly few big chromosomes and many smaller chromosomes. This feature is different from mammals like human or Tassie devil, where we don't have any microchromosomes. They only have big chromosomes. So accidentally, when they were doing research on the embryos of chicken, they discovered that in the subtelomeric region, there are highly transcribed repeats in the subtelomere of chicken, of the embryos of chicken. And for my project, I decided to use these three subtelomeric repeats to map them on the other birds and reptile species. And so the repeats that I've selected, I represent this group, and I also added the telomere sequences to this chapter to be able to look at it in the reptiles and in birds. So the method, obviously, we started with collecting the samples, which Freak always did, and then we, not me. And then I did the cell culture, which I'm very proud of. I did the cell culture, and then we've harvested the cells in their metaphase shape. The metaphase is the best or the highest condensed form of the chromosome, which makes it easier for us to analyze it and look at the different sequences. And then I've used these repeats, the different repeats from the subtelomere plus the ITSs to map them on the lizards and on other bird species. I've used a technique called fish in situ hybridization, which is fluorescence in situ hybridization, which is fish. Probably most of you are familiar with it. I've been talking about it all my project. So the species that I've used 8 bird species, including the chicken, and 3 reptile species. And for the results, so I'll start with the ITS. The ITS, as I said, usually found at the end of the chromosomes. But in all of the bird species and the reptile species that I've looked at, we also found them in different positions across the chromosome. And as the arrows indicate in here. Next, the P041. Some of the bird species have shared similar position of these repeats in the subtelomeric region. Some of them have had the signal in the centromere in the middle of the chromosome, and other had it only on their macrochromosomes. So we have different distribution of the subtelomeric repeats in different birds, but they were still conserved in there. Next one, the CNM. The CNM was only found in other bird species, which is in the parrot species. Usually in the chicken, it's found in the microchromosomes. However, in the parrot, we found it in the subtelomere of the macrochromosomes, which also showing a different distribution pattern. So this is very busy. So this is the summary of all the results from this chapter. And as you can see, no subtelomeric repeats were found in the reptile species. However, they were found in the bird species, not all of them. So this one was found in all bird species, this one in 2, and the last one was only found in chicken. And this just shows the different evolutionary or complex evolutionary dynamic for the subtelomere in birds. And this indicates different time of evolution and the avian lineage. So to summarize all of that, we know now that the ITS can be found in birds and reptiles, can be found all across the chromosome. Second, we know that the subtelomeric repeats were only found in birds. However, so they were conserved in birds. However, the distribution or organization of these repeats were different from one bird species to the other. And now the repeats component, none of these repeats were found in the reptile. I'm so sorry. It's fine. We needed a break anyway. We needed a break. It's fine. That's good, because now I can tell. We needed a break, don't worry. So just the main take-home message from this whole chapter, that none of these repeats were found in the reptile, and this means that these repeats might have evolved in the avian lineage after they diverged from their common ancestor with reptiles. The third chapter, in this third chapter, I looked at the distributions of genes, subtelomeric genes in avians and non-avian reptiles. So for this chapter, I've used the chicken genome as a reference. The chicken have 78 chromosomes. About 9 pairs of chromosomes are large chromosomes, and the rest are microchromosomes. In this chapter, I only focused on the large chromosomes, and I've used that because it's one of the best assembled avian genome assembled so far. So therefore they use it for comparative genomic studies and mapping between chicken and other birds, and sometimes with reptiles. So to do this mapping or comparative analysis, we usually use a technique called or something called BACS, which is bacterial artificial DNAs. So so the BACS usually include DNA from chicken, or the interest of the gene of interest from chicken, or the sequence from chicken, and it's infused in a plasmid, which is bacterial circle chromosomes. After that, they infuse it back in the E. coli. They put it back in the E. coli or bacteria, and then every time the E. coli divide, it repeat or give me a new sequence of the gene of interest that we are looking for. And in the lab here, I separate the chicken DNA from the rest of the E. coli DNA, and I stain it and use it in the fish technique. So for this chapter, as I said, I'm using blocks or the DNA sequences from the chicken, which also include genes within these blocks of DNA. And then I've looked at them in the reptiles and birds. So the method, as I said, we first, in the previous chapter, we collect the chromosomes, and then I use the BACS from the chicken subtelomere, which I just gave you a brief about it. And then these BACS, we stain them with red and green, and I use a fish technique to map these sequences on the birds and on the reptiles. To analyze the images and the signals that we have from fish, I use the Leica fluorescence microscopy. And then at the end of this chapter, I've also used a bioinformatic tool or program called Genomicus to look at the arrangement of these genes and the difference between the chicken and other reptiles and bird species. So the BACS that I've selected for this chapter, from each subtelomeric region, I've selected one BAC, which are distributed at the end in each subtelomeric region, and I've only took BACs from the macrochromosomes. The species that I've used are including 4 bird species, including chicken, and 3 reptile species. To be able to do the analysis for this chapter, I first needed to look at the position of these BACs on the chicken chromosomes. I want to make sure that they are sitting in their position, and then to map these BACs onto other birds and reptile species. So for the first part of this, we've looked at the positions of these BACs in the chicken, and we mapped them on the chicken chromosomes. And most of these BACs, they were found in their right position. However, in 3 chromosomes, we have some interesting results, including chromosome 2, 3, and 6. So I'll start with chromosome 2. So this is the green BAC, which found at the end, at the top of the chromosome 2, as you can see, and then the red BAC from the bottom, it's found in the bottom of chromosome 2. However, we also have some signals, stronger signal here on other chromosomes. What does that mean? So when I blasted, I found it's found in other chromosomes. This means that these different chromosomes share some homologous blocks, which is one of the characteristics of the subtelomeric region. But looking at this one, which is have stronger signal in chromosome Z, I wanted to go further and see why does it have that strong signal. So I looked and compared the genome sequence of this BAC and compared it to the end arm of chromosome Z. And I found that between those 2 regions, none of the genes were similar between those 2 regions, so it's not genes. However, when I looked at the repeat elements in these 2 regions, I found that the end part of chromosome 2 shares some repeat sequences with chromosome Z, and it's called macro Z repeat. So it's a Z repeat, and it's repeated so many times on the Z chromosome, and that's why it was giving us a stronger signal. So pretty cool. Next, chromosome 3 and chromosome 6. So these chromosomes we didn't have a signal and we didn't have a signal in the chromosomes that it's supposed to sit on. So the red one is not sitting on chromosome 3. However, the signal was sitting in the middle of chromosome 1, which is represented in this schematic diagram. Chromosome 6 has similar problems. So the back that we've selected from the top part of chromosome 1, for chromosome 6, it wasn't sitting in chromosome 6. Instead, it was sitting in chromosome 3. So that shows us that the mapping of these or the positioning of these backs on the assembly was not mapped correctly or was not put correctly in their position. Next, mapping these backs, all the backs that we found them in that position, we mapped them on the birds and reptile species. So I'll just give an example from chromosome 1, because just to save everyone time. So that these are the two backs from chromosome 1, which you can find them at the end of the chromosome. And these are the genes that are sitting in this DNA sequence. So when I map them on the bird species, in most bird species, these two signals were sitting on two specific chromosomes, chromosome 2 and 4 in the thornbill and emu, and they were sitting in chromosome 1 in the parrot. So the signal was pretty much conserved. Even they moved to a different chromosome, but it was still there. Now in reptiles, it's a different story. So there was either no signal at all, or some of them had interspread signal on multiple chromosomes, but we couldn't define which specific chromosomes sitting on. We can see it on the chromosome, but we couldn't tell specifically where. So to try to analyze and go further and look at the arrangement of these chromosomes on the chicken and compare it to other species, I decided to use Genomicus to look at these gene sequences. So I just highlighted the one in green and in red to make it easier for everyone to see the arrangement. So for the top one, which is the green gene from the top of chromosome 1, as you can see, we've used Genomicus, which is sitting in that green one, and the genes here. So it's showing just a snapshot of the arrangement of genes in the chicken chromosome from the back that I selected. And as you can see, turkey has the closest arrangement to chicken. However, there is still some inversion and deletion in some of the chromosome here. And the further you go down from the chicken on the phylogeny tree, the genes start to go dispersed and they're sitting in multiple positions here. Because a lot of these species don't have great assemblies, it was hard to decide the exact name of the chromosome. It was just sitting there, but they still show a high arranged pattern in the genes. And then the red one here, same. This is this side showing us this gene, and those are the genes that are sitting next to it in this region. And this one is even more arranged. It was only found in 3, the green one was only found in 3 species, and the rest of the species is just dispersed as well. And even in turkey, the closest to chicken in this phylogeny tree, it was not even there. So what we know from the result that I've had, that the distribution or of the subtelomeric region, it's so in birds it's closer to the chicken, so there is a level of conservation, although they moved and rearranged, but they were still there. In other birds and in the reptiles, some of them they've had multiple signal on multiple chromosomes, or sometimes like the turtle, they were not there, which indicate that this sequences might have been lost in the turtle and they were stayed in other reptiles. So after looking at this chapter, there are 3 main messages that I've learned. The first one, that segmental duplication and repeats play an important role in the subtelomeric region. And we know that one rule of the rearrangement doesn't apply to all. So it's very highly diverse and highly arranged in the subtelomeric region that you can't say one rule is applying to all of them. And then finally, that some of these back that I've selected from the chicken genome were not mapped correctly on the genome assembly of the chicken. Going back to the big picture of the research that I did, just to summarize each chapter, from the first chapter, as I said, I've selected the Tasmanian Devil to represent the marsupials, and I was able to fill all these empty circles. And we know that the general structure between the Tasmanian Devil and the human is very similar or conserved. And then in chicken, from the last chapter, we know now that the chicken chromosomes share different homology blocks between each other. And then in the bird context, we know that some of the birds shared some blocks with the chicken. They shared genes, and then they have conserved the repeats from the chicken to the other birds. In reptiles, we know that some of the reptiles shared some sequences with the chicken genome, and we know that they have they shared some of the genes with the chicken genome as well. So just to look at how did this phylogenetic tree look before and after I did my project. So before, we had lots of empty circles here, which is the knowledge gap. And then after I finished, I was able to fill this part and this part of this phylogenetic tree. So there's still a lot to fill, but I've tried my best to fill these circles. So the take-home message is, we know now that the subtelomere structure is conserved or can be conserved between closely related species, such as Tasmanian devil and human, and such as birds and chicken. They have conserved structural components. However, the organization, the length, and the length of these components can vary between different organisms. And I just want to say thank you to everyone. Thank you all for coming here today to support me. I want to thank my supervisors, specifically Tariq, Janine, and Ambi. And I want to thank Gus and the kids for being so patient with me. Also my family in Palestine, especially my mom and my dad. I want to say thank you to all of my friends here at UC and the people that left already. I want to say thank you to all the PhD students that are sharing the office with me as well. And I want to say thank you to all IE and SciTech staff, everyone, professors and postdocs who's helped me a lot, and lab managers, Barbara as well, everyone who's helped me and supported me in every way. And thanks again for all your help and support. Oh, did I know?
So in the segmental duplications that you noticed in the subtelomeric region, were you able to determine if those were containing coding genes, or were there non-coding genes in there that were silenced, and what role is that playing? Do you know?
So the segments, we call it a segment, duplicated segment, if it's bigger than 1 kb up to 200 kb. So obviously they have genes within them, and the genes can be duplicated. But the ones that we looked at in the chicken genome, especially between the Z and the chromosome 2, I couldn't find any shared genes between the two regions. It was mainly, so I blasted every single gene from the chromosome 2, and I couldn't find any of it in the chicken, at least that is found in the genome assembly so far. But they shared repeats, which are common between these two regions. But in theory, they should duplicate the genes as well.
Yeah, great talk, really well presented, and
Thank
great,
you.
great body of science. So it's fantastic to hear it
Thank
all come together. I just had one question that could be coming from my ignorance, and I'm not going to mix up COVID and Kittery in my question. But basically, yeah, with the repeats. So given that a lot of your signals are based on repeats, how certain are you that you're looking at homology as opposed to convergence, because the repeats can obviously reoccur a lot easier than other structural elements within the genome?
So in the I looked at the repeats in my first chapter only. The second chapter was actually blocks, which can include both genes and repeats. But the second chapter, which is this one, so those repeats from the pattern that at least looked at, some of these were found in all chromosomes, and some of them they shrinked into two in the chicken and the parrot. But when they only were found in the chicken and the parrot, as you can see, the chicken and the parrot are the closest in the phylogeny tree. So we don't know, sometimes these the nature of the segment of the repeat elements, they can shrink and then re-expand. And but from the ones that I've looked at here, the PO4 was found in all of them. Some of them they had similar to chicken, and some of them had had less signal. I was only in a couple of microchromosomes, so they didn't disappear 100%, but they shrunk. And then in the CNM1, it was only found in the parrot, but the signal in the parrot was even bigger than the signal in the chicken. So it expanded in the parrot, but it didn't in the rest of the birds. Unfortunately, in none of these repeats where I couldn't find any signal in the reptiles, so I can't say if they were there but they shrunk or they were just so. And that's why we said they might have evolved in the avian lineage after they diverged. But it doesn't mean that there isn't repeats in the other reptile, and similar repeats, because some of these repeats they belong to big repeats families, and they share some identity with their family. So it might be just a different component. The PO4 is, this CNM1 in ducks. It has a similar one, but it's not the same repeat, but it has a very similar from the same family, but it's not the same one. So it might be just a different repeat, or different from the same family, but at a different repeat. I hope I answered your question.
Yeah.
Yeah.
Thank you. Oh, awesome talk. Good fish pictures. Really lovely. I just wanted to ask, like, combining your chapter 1 and 2 and 3. So chapter 1 you did bioinformatics. It's okay, you don't have to go. Bioinformatics, and then you applied on chromosomes, and in chapter 2 and 3. Now, in chapter 1 you used marsupials. So based on everything, if I replicate that analysis of chapter 1 in chicken chromosome, do you have hypothesis that I'm going to get the same kind of results or different?
So in the chicken, I didn't do the bioinformatic analysis.
No. If I do it, like, what is your opinion? Like, it's going to be same, or?
I say, yeah, I should have mentioned at the end. So when we looked here, we can see that there are some conserved components of the subtelomere across from yeast to parasite, including different amniotes. So the homologic blocks, they were conserved except in insects. The genes, they were there. But however, as I said, for the chicken one, there's still some gaps. But for the ones that we've looked at, we can say that they are conserved between the human and the chicken. but we still need to fill these gaps. That's another chat, bioinformatic chat. Online. That's Fiona. Yeah, thank you, Fiona, for your question. So Fiona's asking, what does it mean if some species have ITS, a lot of ITS, and some species don't? Generally, as I said in can we get rid of that? So these repeats that I just wanted so picture of So if you look at the repeats in the chicken, they have so much of these ITS spread all across the genome, but in other species like reptile, they don't have as many. Generally, in the term of the evolution of these repeats, it means something, but in terms of the function or the effect on the gene expression, I'm not sure if it has any implication on the gene expression, but they are used to look at the evolution of the genome from one species to another. So they've noticed if it's spread in a lineage, it actually have similar pattern. And if it starts shrinking a lineage, it has similar pattern. So it's mostly used for to predict the evolution of the genome from species to species. And also one more thing that I wanted to say, that in the chicken, they've noticed that just not comparison between two organisms, just in the chicken itself, they found that these ITS are more concentrated in the microchromosomes. And they think that they sit in the microchromosome to protect these genes because it has high density genes in the microchromosomes. And then the aggregation of the ITS in the microchromosome is related to the high density of genes to protect them or silence the genes in this region. Any other questions?
All right, there are no other questions, so please let us put our hands together. If there are. Vicky and Rod for again organizing Characterisation, and we'll be going to go for lunch. So to Yum for the end, I think. So please do with the guys. Thank you.
That's fine. That's great. No,